So first of all, I feel bad for myself following Rui, so everybody just kind of bring your expectation levels down. This is a weird world to be in. Thanks for inviting me here. It's been a while since I've talked about flow. I've been out of the flow market for a little while. What I'm going to be presenting is not something revolutionary or an advantage that we have with our microfluidic device, but a technique that I've seen gain popularity over time. And that's about sample prep for nuclei. We don't enable it. You guys can do what we do. You do it faster, but the process is the important part of it. I see a lot of friendly faces here that I've known in the past: Mike, Daniel, others, Casey. I took a couple years off, worked for Beckman and NonFlow, joined NanoCellect this year. I still haven't quite got used to this virtual world, even though I've been doing it for 19 months. Hopefully these things will go away and this will be stories we can tell our grandkids. What I'm going to be discussing today is a nuclei sorting method called Frankenstein. My original title of the talk was going to be Frankenstein Meets the Wolf, but I thought better about that. Unfortunately, I need to go over a couple slides that are going to look like they're marketing heavy, but since what we do is low and slow and what you do is high speed, I just want to show off some differences. I need to adjust some of the marketing language after what Rui showed, so I'm going to have to borrow the slides, Rui, and fix some of our content. Whoops, I missed that one. What we have are two platforms, the Wolf and the Wolf G2. So the things that they like to talk about here is high viability. Rui just pointed out that slow doesn't also correspond to high availability. The advantage that we have, he pointed out in his last slides, are sterile and disposable. There's no contamination. We're not generating aerosols. Everybody says it's simple, intuitive. We do have a small fit, so if you want to put it in the hood, it will. What I'm going to be talking about today is using the Wolf in the Frankenstein protocol. What's not shown is a chiller that we attach to it, and I'll point out why chilling is very important in the nuclei prep. I'll have to admit, I joined this company. I've been here almost 9 months. It took me 2 months to realize that the name of the instrument is flow spelled backwards. So a lesson learned. What our advantage is, or what we think our advantage is the microfluidic chip. Everything is done on here, meaning once you're done with it, you throw this away. There is no cross-contamination. There is no aerosols. Everything is done within this chip. We have 3 ports here. We have A, B, and C. A sort to the left, B sort to right, and B goes right down the middle. You can collect it and reuse it if you want. This is just an animation showing the flow, and we use a piezoelectric activator to push the stream either left or right. Unfortunately, in this slide, I see that I call it A, B, and C, and they call it A and B, but it's A, B, and C. We are slow. We don't go very fast. We're low PSI. We sort only about 200, 300 events per second, but it has enabled us to get in some worlds that are new for me. For instance, we spend a lot of time in cell line development, partly because we have a piece of the instrument called the N1 that enables us to sort into 96 well or 3 to 4 well plates, assuming you can find these plastics. They're on back order forever, I think. Genomics, gene editing, immunology, and infectious disease. Because it's so small, will fit in the hood. What I'm going to be focusing on today is this genomics workflow in the chromium assay. So this is the workflow. It doesn't mean that you couldn't put your instrument here and do the same thing. I'm just highlighting what it is. Nuclei, apparently very difficult to deal with from a reproducibility standpoint. I've talked to other core lab managers who dedicate an instrument just to nuclei sorting to get rid of the variability, because contamination and cross-contamination and instrument variation is a problem. This method doesn't come from me. It comes from people much brighter than me. It comes from Luciano's group. If you don't know him, he's a scientist director of single cell core at Harvard. Working with nuclei has its own challenges. They're fragile. Isolation methods can be too harsh, low quality. And once you have garbage in, you're going to get garbage out. This method is becoming popular just because it's robust, reproducible, and the name comes from splicing 5 different experiments together. Therefore, the name Frankenstein came out of it. What I'm going to show you is a nucleation process paired with a wolf. It doesn't mean it can't be paired with anything. The reason it's gaining popularity is because the protocol has been demonstrated to work successfully with frozen tissue, cryopreserved cells, and cell lines. Typically, when you work with a nuclei prep, it's going to be geared to your cell type. In addition, this works with a lot of diverse cell lines, including solid cancers, pancreas, paragangliomas, breast cancers, lymphomas, xenografts, and tumors. It does not work with cardiomyocytes. The myosin heavy chain is just too rich in that sample. So this is really a sample prep machine where we're going to get rid of garbage so that when we get to this point and we do the chromium assay, we're ready to go. Now I lifted this from 10x's website because it's very telling why assays go bad. In this case, A and B show that the nucleus is relatively intact, and C and D are horrible. I can tell you, if you want to recreate D, keep your samples at room temperature. You'll get that every single time. The Frankenstein method. It takes about 30 minutes to go from beginning to end. So there's tissue homogenation, which is basically slice and dice with razor blades, nuclear isolation buffer and staining, and then cytometry. What I'm not going to show you is the contact point between us and chromium because we don't have a chromium system, but this is about a 15-step process. We got mouse brain cells from Rockland. You can get them anybody you want, but it's easier to do that. The buffer of choice is really not important. This is what we used. They described 2 other buffers you can use, but this was off the shelf and we didn't have to recreate it. Nuclei were filtered and everything was remained at 4 degrees C during centrifugation. And keeping things cold is critical. In our system, we didn't keep them on mice because we do have a chilling unit associated with the wolf. Nuclei suspension was also stained with 7A-AD to delineate debris from nuclei. 200,000 nuclei were sorted. What you can expect in this experiment, not just because it's our instrument at all, but about a 50 to 70% loss of nuclei when you go through this process. Here's the data that we all like to see. This is pre-sort. We have a different way of doing of getting singlets. It's because we don't do area. This is our singlet population. And this is when we stained with 7-AD. So you can see there's a ton of debris here. And in this picture here, we stained with trypan blue so you can see the nuclei and all the garbage associated with it. After sorting, we enriched it to 87%. You can see that everything's intact and pretty well clean. Now, what we're missing is the punch that says this also translate to a 10x good assay, but I think we can all take that leap of faith at how we got there from there. Now, Rui, I'm going to give you some time back because this is my last slide. So the wolf cell sorter is a gentle microfluidic sorter that can gently and accurately sort nuclei while removing the tissue and debris. After sorting, we maintained high quality nuclei, which demonstrates its capability of performing well on 10x assay. I put the citation in here. It's nice to go to the site because it's almost like at the Purdue user group, you have other people commenting on things that work well, things that don't work well. For instance, the 10x world says you can only use 7-AD, but the cytometry world says you can use PI and DAPI with no problem. That's it for me.
Okay, thank you so much, Robert, for that wonderful talk. I actually have a question about the platform. I'm curious to hear if there are any plans or if you know of any development being done to see if there's a potential for the sample input mode to be adjusted, right? I think it was recently a question on the Purdue list where someone was asking if instead of loading 5 mil or 15 mil tubes, and I know the Wolf allows for 50 mil as well, if you could do plate sample input. Is there any plans for that?
Yeah, there are. Yeah, we've heard that comment too. So yes, there'll be a plate in the future. We're also working on other things to help with the problems with the wolf. One of the things that we do is, we're not doing droplet in air. We're kind of moving a slug of fluid either left or right. And as a consequence, we dilute over time. We're working on a project called Cluster that's going to be able to concentrate cells and release them in a known batch without having to centrifuge. The other thing that we're working on is image-based cell sorter. So there's a lot of new stuff coming out of this company.
Wonderful. Another question I have is if you know of any facilities that have incorporated this into like a fully automated platform for cell culture, right? Where you might be able to have kind of like a full automated system for sorting and then culturing or downstream applications.
Yeah, there's some companies that have got a leap on that process. If you look at a company called Cytina, they have their single cell dispenser in a hood with a complete workflow from beginning to end. I think that's a little ambitious for us. I think what we're going to spend our time and effort on is it's a good idea. We're just too late to the game. We're going to spend our time and effort on the image-based cell sorter.
Wonderful. That sounds really exciting. Okay, awesome.